So this lightning talk is for people that want to find their match. But when I mean match, I really think of a collaborative match. So the whole point, as Sarah was saying, of ResearchBazaar is to try to bring people together that might have similar research interests and then potentially ignite new collaborations. And so if you're still looking for a special someone to write scientific papers with, discuss about science all day or part of the day, to submit grant proposals with, collaborate on cool projects with, or complement your research, then this is a good moment because we have created what we call a collaboration compatibility questionnaire. We are matching people that produce methods from data science, so data scientists, with people that have data. So if you fall within any of these two, you're a domain scientist, a data scientist, you should go to the QR code right now. Actually, I'm going to pause for people to go to the QR code. It's a very short questionnaire, so it should take you less than 3 minutes to fill it out. And what's going to happen is after the QR code is going to stay there, so I'm going to pause because this talk is actually going to be less than 3 minutes. But in the next slide, we'll still have the QR code up there. Okay, so very smart scientists from the Wisconsin Institute for Discovery will be applying sophisticated matching algorithms to find your perfect collaboration match. This is 100% guaranteed. And thank you, Michael Ferris. Where are you? Oh, there he is. So please fill out the form. If you're interested in participating in this collaboration match, this is the first time we do it, but we are guaranteed 100% find your match. Please fill it out by 11 a.m. today. And then join us at the lunch at noon at the Orchard Room. So you're going to pick up your lunch box and then walk to the 3rd floor. The elevators are by the Aldo Cafe. And then there you will find your match among all the people that fill out the questionnaire, obviously. And then we'll have time during lunch for talking to your match and then other people that might be similar interests to you. So this is my last slide, I think. Yes. So I'm going to give you 10 seconds for people that will still want to check out the QR code, and then that's all that I have to say for today, and I hope to see everybody here in the collaboration matching lunch. Thank you.
Hello, everyone. Thank you for inviting me to participate today. I'm going to be talking about NVivo and how to use that application to manage and interpret qualitative data. My name is Tom Durkin, and I'm with the campus library system. So what is NVivo and what is it used for? NVivo is an application that can be used to manage and analyze qualitative data. I'll talk a little bit more what that is in a minute. NVivo facilitates the storage, organization, and categorization of project documents. And NVivo will save a researcher time by automating qualitative data analysis tasks, such as coding text. You can get the NVivo application through the Lumivero company. If you have access to the campus software library, you can download that application from there too for certain campus people, not everyone. So you have some data. Is it qualitative data? Qualitative data is frequently text, such as interview transcripts or field notes. It can also exist as images, audio, and video. Qualitative data is gathered by researchers making observations, conducting interviews, doing surveys, and collecting documents and media. So what is qualitative data not? Qualitative data is frequently defined in contrast to quantitative data or numerical data, so it's not numerical data. We're mostly talking about text. So what types of analysis can we do in NVivo? Excuse me. Fundamentally, NVivo is a tool that will help you to deeply read, understand, and interpret data such as textual interview transcripts. We usually start out by coding. So coding is marking sections of a text with thematic tags to identify themes. You can see there's sort of an example there. Some types of analysis you can do include word frequency, which words occur most frequently in a set of texts. And I've got like a word, kind of like a word cloud there. Words in context, what phrases are associated with a specific word, and we have another visualization you can do there, a word tree. And then there's a matrix coding query you can do to see how frequently different coded themes overlap with each other, the co-occurrence of different themes. And these visualizations are also part of the NVivo system. So I am here to offer training and assistance. I offer NVivo training classes through the OHRD system. I schedule individual NVivo training sessions, and I can also assist with troubleshooting. You've got my contact information there. Please let me know, and I have some handouts for everybody if you visit me at the table in a little bit. All right.
Hello everyone, I'm Yuchen, a CS PhD student working on large language models and machine learning fairness. So today I'm going to talk about my recent work on large language models. So I believe everyone has already heard about ChatGPT. It has been very popular recently, but actually before the ChatGPT shows up, the GPT models has been very popular family of sentence generation language models. They have been known for its zero-shot, one-shot, and few-shot capabilities. So zero-shot means like we don't need any data at all. We just provide a task description, and it can be used for certain tasks. And one-shot is like we provide one more example there, and then so that the language model can understand the task better. And few-shot, like we provide even more examples. And here, actually, we focus on the last scenario. Just we provide a small data set to fine-tune the language model for certain tasks. Just like provide a lot of data. So we know language models can understand the task context, but unfortunately, in most machine learning techniques, we get rid of the task context at all. For example, for the iris classification, I think most of you have already been very familiar with this data set. So just give you some petal length, petal width, and the task is to predict the type of this plant. However, in most machine learning tasks, we just get rid of those color names and do some one-hot encoding to convert those categorical features into numbers. So finally, we only use a bunch of numbers instead of the task context information to make the prediction. So task context information are not used at all. So to address this issue, we just use language model for solving those machine learning tasks. For example, so what we do is we just convert those numbers into sentences. So instead of directly input those numbers into the model, we just first convert into a sentence. Like here is an example for the iris data. So instead of just use the numbers, we describe it as an iris plant with sepal length 5.1 cm, etc. And then we provide the true class at the end. So those are examples we will be provide for this model to understand the task context. And then after fine-tuning the model, for solving this task, now give a test data, which we know the sepal length, sepal width, etc., but we don't know the true type of this plant. So we can still just convert it into a description of the feature. And then we just ask the language model to complete the sentence. So this is what language model do. And we found actually it can recover its true label, true class of the plant. So this is the basic idea of our project, and actually we found it is very powerful and it works for a lot of different tasks. So I didn't show any experiment results here, but yeah, if you are interested, you feel free to ask me questions after my presentation. Thank you.
A lot of health science uses hypothesis-driven research, meaning there's a research question driving the science. The Survey of the Health of Wisconsin is the opposite. We use non-hypothesis-driven research to foster collaboration across many disciplines to advance health research in Wisconsin. On average, it takes 17 years for biomedical research to translate into action. SHOW can expedite this process through its ready-to-use data, biospecimen, and research services. To date, SHOW has enrolled more than 6,000 participants from across Wisconsin, representing the general population. Most of our participants have consented for their data and biospecimen to be used for future unspecified research and have agreed to follow-up studies in the future. Together with our survey data, the physical measurements, biological and environmental samples from our participants can connect what happens inside the body to social and environmental factors outside of the body. Additionally, SHOW can link participant data with external sources of data like GIS and the State Cancer Registry. Over the years, these unique analyses and linkages have created additional data sets available for students, scientists, and practitioners to use. These existing biological samples and data sets help scientists answer their research questions in a faster and more affordable way than starting from scratch. The SHOW Research Services platform supports investigators from diverse disciplines to launch both prospective and retrospective studies. Retrospective studies are built on our existing data and biospecimen, while prospective studies often recruit SHOW participants for future studies or utilize our infrastructure to collect new data. Because SHOW is not hypothesis-driven, we are able to collaborate with researchers across campus, including investigators from bioinformatics to nursing to pharmacy to kinesiology. A current retrospective study by Dr. Mohammed Murtasa is analyzing urine samples from 65 seemingly healthy show participants. 13 of these individuals received a cancer diagnosis within 6 months of donating the urine sample to show. Dr. Murtasa's assay can detect multiple types of cancer in urine. With the help of these show samples, we'll learn just how early, prior to a clinical diagnosis, this test can detect cancer cells in the body. And a final example, a recent prospective study led by Dr. Janine Dilworth-Bart featured collaborations among epidemiologists, biostatisticians, toxicologists, engineers, and child development experts to investigate cumulative exposures among preschoolers. So 11 kids between the ages of 3 and 4 years old completed the study, which included wearing an air pollution monitor outfitted in a backpack, a word noise detection device in a shirt, and donating numerous biological samples and completing questionnaires and cognitive tests. While this was a feasibility study, collecting all of this data simultaneously will help researchers understand the relationships between chemical stressors like air pollution, non-chemical stressors like noise, and psychosocial stress like parenting quality, and how they might impact academic readiness and health outcomes. SHO was well positioned to facilitate this study because of our expertise in piloting new data collection methods and background with community-based recruitment. These are just a couple examples of how SHO was able to foster cross-disciplinary collaborations to gather data and expedite the impact of biomedical research. How might SHO support your research?
Okay, hello everyone. My name is Zhenmei, and I'm computer science PhD student. My advisor is Professor Ying Liang. Today I'm going to review the neural network learning from empirical and theoretical perspective. So as we all know that in 1986, Geoffrey Hinton proposed backpropagation in neural network. And in 2012, Hinton and his student Alex proposed that AlexNet, and it gets the championship in the image classification competition. And the AlexNet has 8-layer neural network. And then in 2014, we have VGG from Oxford University. And VGG have more than 20 layers neural network. And then it's a big milestone. In 2015, we have ResNet proposed by Khine He. And ResNet is the first model beat human in image classification problem. And it gets a champion in that year. And then it's 2016, we have AlphaGo beat one of the best Go player in the world. And then it's BERT. Then we have like attention all you need paper and transformer structure. And then in 2020, we have GPT-3 from OpenAI. And also in 2022, we have November, we have ChatGPT. And most researchers believe that ChatGPT is very close to artificial general intelligence. So OpenAI also said that maybe in this year, April or May, they will release GPT-4. And it is some researchers believe that this may be the first model achieve artificial general intelligence. Then what is the progress for the theoretical analysis? So it is much far lay behind the empirical one, we can see. In 1994, we studied linear model, just one layer. And then in 2007, we have random feature. It was very similar like linear probing nowadays. We just train the last layer, the top layer. And after ResNet, lots of researchers believe that neural network is very powerful, and they try to study that. Then in 2016, we have deep linear model. We do not have any activation function, so it is still can only handle the linear separable data, but it is already very complicated. Then in 2017, we have mean field analysis. We see the neural network as a PDE method and trained by PDE and analysis the continuous gradient descent. And in 2019, we have neural tangent kernel. We see the neural network as a kernel method. And if it has a large enough initialization, its training is very lazy and it stays in its neighborhood. We can just consider the first Taylor expansion of the neural network. So we call it kernel method. But after that, lots of work believes that the neural network is beyond NTK because they have feature learning. So lots of work use different methods to show that neural networks is beyond NTK. And our work shows that, okay, we consider general input data and we propose a general Green feature learning framework. And it is maybe beyond all the previous work. And I hope our work can make a little forward step for understanding neural network. And if you are interested in the detail, you can come to discuss me in the discussion section. Thank you.
Okay. Hi, I'm Trisha Adamus. My pronouns are she, her, hers. I'm a research data librarian at Ebling Library in the medical school. I'm here representing Research Data Services, of which I am consultant there. I would like to mention that Research Data Services, if you're unfamiliar with them, does three main things. We do help with data management plans, which is going to be the focus of this talk today. We also do personal consultations, so we can meet with your lab group, we can meet with you one-on-one and talk with you there. We also do education and training, much like this, so we can do presentations, and we also do fun activities. We have a reproducibility activity involving Legos, so if that sounds fun, contact me. So my main question for you today is, are you ready for the NIH's data management and sharing, or DMS policy? If the answer is no, that's okay. I'm here to help with Research Data Services, and I'll talk to you a little bit about that. So this is a slide that I have lifted from my talk that I give. I've been giving it since July, but if you want to talk more with me about it, great. This is just very generally what you need to know about the policy. So the policy is in effect now because it started after January 25th of 2023. It is for competing applications. So when you go to put in a proposal, you are going to have to submit a 2-page plan outlining how scientific data from your research will be managed and shared. This is going to potentially change your workflow as researchers because when you get an award, you are going to have to share the data, and that data should be shared as soon as possible, which means no later than the time of an associated publication or at the end of the performance period, whichever comes first. So if you are going to be publishing during the middle of your award, that's when you're going to share. If you haven't published yet and your award is done, that's when you're going to be expected to share. So just so you know, this is coming. And I will move to the next slide. We have this slide available on campus. We have something called DMP tool, which will help you manage your data. There are 6 elements in the data management and sharing plan offered by the NIH. The DMP tool will walk you through that. It'll provide you guidance with what is available from the NIH and also what is available here with UW-Madison. When you get past that, you receive your funding. When you get to the end of your award period and you're going to share, there are options for generalist repositories here on campus. The top 2, Dryad and Minds, are for open data that is non-sensitive. Dryad, we have a membership, and you can deposit unlimited data up to 300 gigs per deposit, which is great. Minds at UW is our institutional repository, and the next 2 are for sensitive data. I would like to mention that we give this talk we give the larger talk every Wednesday, and we have open office hours every Tuesday. So please find us. That's it.
Imagine teaching a classroom full of children when you suddenly hear aircrafts flying overhead, dropping bombs in their wake. You frantically gather your students to take cover, but before you can do so, you see soldiers marching into the classroom, shooting at anyone in sight. This is a daily reality for children and teachers across Myanmar, a country in Southeast Asia roughly the size of Texas, with a population of about 54 million. Despite the bloodshed caused by the military brutal crackdown of pro-democracy activists there, it has received very little international attention. So today, I'm going to introduce you to key issues in post-coup Myanmar using a news headline analysis. I used a Burmese news site called Myanmar Now to scrape news headlines from 2021. And to get a sense of the most common words in these headlines, I made a frequency dictionary. I saw that words like Mandalay and Sagaing, which are circled on the left on the frequency dictionary here, denoting regions in north-central Myanmar, as you can see on the map on the right, showed up as the most common words, along with words like junta, military, soldiers, and resistance. This implied to me that reporting about military attacks and civilian resistance seem most associated with these regions in Myanmar. But this frequency dictionary didn't give me a sense of distinct topics that were reported because there were many similar sounding words here like junta, military, and soldiers. So to get a sense of distinct topics, I turned to article tags that the journalists themselves use to categorize each article differently. And due to runtime issues, this time I limited my analysis to articles from 2022. This graph shows the most common article tags from articles in 2022. The top two most common tags were armed resistance, which is the blue line, and Zeng region, one of those states that you saw in the map earlier, the red line. So we can tentatively see that in 2022, like in 2021, military attacks and at least the armed resistance seem mostly again in the north central region in Myanmar. But what about the military attacks that we saw feature so frequently in 2021 headlines? What I've highlighted in black here is the trend of the tag military raid. As you can see, it declined significantly after April, as opposed to the tag, the blue line, armed resistance. Now, this suggests two possibilities. One is that military attacks really did decline in 2022, or the journalists are making some kind of editorial decision about what to focus on. Perhaps after 2 years of constant assault, they want to raise the morale among their readers and want to focus more on continued resistance rather than on continued oppression. And that's one of the questions I want to keep exploring. So if you are interested in this topic, please come talk to me at the discussion. Thank you so much for your attention.
Great, thanks everybody for being here, and thanks to the organizers. So I'm Deshpande, I'm an assistant professor in statistics, and the method I'm going to talk about today starts from this very kind of general problem. So we're going to imagine that we have a network. This might be a social network where the vertices are people and the edges are like friendship relationships, or maybe the network is, the vertices are spatial units like census tracts, and the edges correspond to spatial adjacency relationships. And the setup here is that at every node in the network, we're going to observe some XY pairs. We're going to observe some predictors and some responses, and we'd like to do regression. We'd like to, map the predictors into the outcomes, and we'd like to do this at every vertex in the network, but we don't want to assume that the regression relationship is the same at every vertex in the network. So as one example, if you're modeling crime in a city, maybe you measure crime at a neighborhood level. Maybe there are some predictors that you can measure at the neighborhood level, but the temporal evolution of crime is not the same in every neighborhood across the city, but maybe it's similar in neighborhoods that are next to one another. And so we can frame this as a regression problem where we've got some noisy observations of a function that takes as inputs your covariates or your predictors X and the label of the vertex in the network. And we'd like to learn this function in as flexible of a fashion as possible. We don't want to make parametric assumptions. We don't want to make really strong network smoothness assumptions. We would like to also learn this function in a way that is conducive to good uncertainty quantification. And this seems like a tough problem, and it is, but I think we have kind of a cute solution. It involves approximating this function as a sum of regression trees, which leads into its own problem of how do we define regression trees over networks? How do we incorporate network structure or structured variables into regression tree ensembles? And it turns out we can do that by being very clever about creating some new stochastic processes to recursively partition networks. And so one example is maybe you have a network, maybe these 10 nodes, maybe I draw a spanning tree at random, and I delete a random edge. And this will produce a bipartition of my network into connected components. So this is just one example of ways that we can split on a network label in regression tree ensembles. We've implemented them in a new R package, and so I'm very excited to chat more about it after this or over lunch. And I want to thank everybody for listening. Thank you.
Hello, everyone. My name is Ting. I'm a second-year CS PhD student. Today, I'm going to present my recent work called Active Cost-Aware Labeling of Streaming Data. This is a joint work with my advisor, Prof. Khine. So first, our motivation comes from the fact that machine learning nowadays is very data hungry, but labeling data can be very expensive. So let's start with a very concrete example. Suppose you're a gold digger and you have already drilled at all the black points, and you know that the blue circle indicates there's no gold underneath it. Now your manager tells you you have new 2 spots, the green star and the red star. If you are the gold digger, what would you do? Considering the fact that operating a drilling machine can be very expensive every day, sorry. So you probably don't want to label the green star because you have already explored all its surrounding areas and it has a very low probability to have gold beneath it. So the good choice is definitely to label the red star because we have very limited information around it. So our goal in this work is to trade off between the prediction error and the labeling cost and learn how to label data well. The difficulty of this problem really lies in the tension that if we choose to label, the prediction error will be very small, but the labeling cost is very large. And if we not label, you will get very low cost, but you might have a very large prediction error. So in our paper, we have considered this problem in two settings, the discrete setting using multi-unbounded and a continuous setting using Gaussian process. And surprisingly, we have found a method that is proved to be very powerful in both of the settings. The strategy is very simple. It's just that when the data comes, you always choose to label it if your uncertainty is larger than a specified threshold. And in our work, we have proved that this strategy is actually the optimal strategy in both of these two settings. And we also compare our method with 3 other baseline methods on the right. As you can see, the red line is our method. It outperforms all the other 3 methods by a large margin. So that's all for all my presentations. I have hide all the technical details and just try to give a very concrete example. If you have any questions, feel free to ask. Thanks very much.
Hello, my name is Henry Hundt. I am a master's student in the Energy Analysis and Policy program, and I work in the Climate Action Lab with Professor Morgan Edwards in the Office of Sustainability, where we are trying to find the correct path forward for deploying renewable energy at scale for UW-Madison so they can reach their clean energy goals. But as a byproduct of this process, we're trying to make this data available to address one of the biggest issues in renewable energy development right now, and that is finding land where there's a community that wants it. And this is not only a big issue here in Wisconsin, but also very broadly across the United States. And our possible solution to this idea is to make the data for siting renewable energy as public as possible. And this is because we believe that communities in general want to adopt clean energy, and that they want to do it in a way that fits into their understanding of their land use and so forth. In Wisconsin, this is specifically a hot topic because of people's association with land as being agricultural productive, even if they're growing products that might be eventually used for transportation fuels. So the goal here is to, as we go through the development of these layers for all of UW-Madison properties that are outlying outside of Madison, we're making those layers available without any sort of optimization or anything so that they can see what is the kind of general value of a parcel that they might have their eye on. And for example, we're doing one of the layers that we developed was just doing a basic slope analysis of property of all land across Wisconsin and showing where PV installation is viable for like a community scale system. And in doing so, hopefully we can enable this sort of faster, more quicker development of renewable energy in Wisconsin, and also get out ahead of some of these challenges that the university will face when they try to develop renewable energy projects in other parts of the state. And the goal is also to use satellite data as often as possible because we want this to be something that can be developed outside of the United States as well. And if we can use the more macro scale data we can use, the more likely that it will be able to be reproduced somewhere else in the world. Thank you.
Hi everyone. Today I want to share with you a method to infer phylogenetic networks. On the left is a phylogenetic tree. It can capture the biological evolution, but it is unable to capture some important realities like introgression, hybridization, or like gene transfer. So on the right is a phylogenetic network. This one is able to account for reticulate evolution, and this is an extension to the tree method. But all existing network methods nowadays are unable to handle big data set. It requires the search of the network space and perform numerical optimization, which can be super high time consuming. So our method is able to reconstruct network based on algebraic invariance, which can overcome all the problems I mentioned earlier. Our method only requires the evaluation of polynomial equations, and this is able to avoid the search of the network. It also avoids optimization, which can save a lot of time. The workflow of our method is shown down below. We begin with a concordance factor table. Each concordance factor represents a proportion of gene trees that exhibit a certain structure, and all of these values will be mapped to a vector. This vector will be imported to the invariant functions for each candidate network. And in the end, the candidate network with the smallest invariant score is going to be selected as the best network that best matches with our input data. Our experiments have shown that our method is more than 10 times faster than all existing methods. And under all simulating scenarios, our method is able to select the correct network with the best accuracy. Our paper has been published to arXiv, and the Julia implementation of this method is available in PhyloDiamond. So if you are interested, please go and take a look. Thank you.
Hey, good morning, everyone. My name is Durkin, and here today I'm going to tell you a little bit about our ongoing research project with my advisor, Alberto Del Pie. So the topic is solving sparse PCA via semi-definite program. I will talk to you a little bit more on the robustness and also the approximality of the algorithm we gave. So we start by giving an introduction of what the problem is. For those who are not very familiar with PCA or principal component analysis, the task is essentially trying to find out a linear development of a given data set. So this is a figure that I found from Wikipedia. And as you can see, maybe this sort of line is the one principal component that we're looking for. It has many applications, including clinical analysis, genetics, computer visions, data minings, and so on. But what we're doing here is that we're focusing on a sparse version of PCA. The reason is that the task is essentially finding a few variables that try to characterize the linear development of a data. And the advantages are quite clear because we're reducing the features out of maybe hundreds of thousands or even millions of features that we collected at hand so that we can reduce the hardness for us to understand what is going to be the key contributor of the data set. And also it has many other advantages, including avoiding overfitting in machine learning. It has been shown there's some results on that. But I would also love to point out that this problem is fairly hard, not only because it has an exponential size of the candidates for those few features we have, and also the problem is non-convex. So it's never going to be easy for us to find out the optimal solution. As a theoretical guy and also an optimization guy, I am interested in finding out a solution or approximate the solution in polynomial time. And recently, it seems like semi-definite programming is becoming more and more popular for solving those sort of problems. So we also would love to see what's going on because there are so many things that have not been shown in the literature right now. What we have now for a result is that in a particular statistical model where a hidden sparse PC exists, but we don't know what that is, our algorithm can find at least approximate the true hidden sparse PC fairly well, even at the existence of some modifications to our data. Such modifications can include noises like Gaussian noises or mistakes when we're collecting the data, or even someone maliciously trying to contaminate our data to some extent. Of course, this problem also has many drawbacks, so we're also wanting to fix in that. So I will put everything detailed in the discussion session so that probably you can chat with me. Thank you very much.
Hello, everyone. My name is Jay Winston. My pronouns are they, she, and I'm a graduate student in educational psychology and learning analytics. So, as you all know, we live in a data-driven society, and we use data to inform our decision-making every single day. But I have a question. Can we actually say that we're making data-informed decisions if the data that we're using is not reliable or valid? I mean, let's think about this for a second. Would you take information collected about an apple to make a decision about an orange? I mean, I would hope not, but hey, everyone has a different shopping strategy. All jokes aside, I can imagine this may be the case for many industries. However, I can assure you, these types of questions are being addressed within academia with learning analytics at the forefront. So what is learning analytics, you ask? Learning analytics is focused on generating actionable data intended to improve teaching and learning by informing the structure, the content, delivery, and the support of the overall learning environment. And as a learning analyst, I've discovered that measuring and collecting the right data points about learners and their experiences is just as important as the analytical methods that we use to gain insights from that data. If anything, what and how we collect data greatly determines our analytical methodologies. And this type of assessment strategy is integral to the continual iteration of your learning experience. For example, we can use supportive data to determine how a learner engages with materials, how learners are engaging with each other, how learners perform on some sort of targeted outcome, either before or after some sort of intervention, and so on and so forth. All of these data points can be used to iteratively design and improve elements of your overall learning experience. Now, on the other hand, your instructional design plan provides critical information that contextualizes and gives meaning to your learning analytics data. For example, without an understanding of what learners are doing in an educational environment and why they're doing it, the resulting data will hold little explanatory power, which circles me back to my main point, the importance of measuring and collecting the right data points about learners and their experiences from the beginning. The collection process informs the analytical methods we use to gain insights from that data, and this type of alignment is critical if we want to truly and confidently say that we're making data-informed decisions to improve teaching and learning, and overall improving student outcomes as well.
Hi, I'm Michael Lade with DeWitt's Research Cyber Infrastructure team. And today I'm going to talk to you about data management plans and some of the options that are available for campus to help. To build on Trisha's talk from this morning, that data management plans and sharing have been a really hot topic on campus all over. With NIH, their policy going live this year, now all the major funders are requiring data management plans up front at grant proposals that'll highlight how data will be managed and then published at the end of the life cycle. So today I'm going to highlight a couple of tools that are available. The first is ResearchDrive. It's a university-wide file sharing service that's available to all PIs and their research groups. It's a general purpose file storage service, and it's used for a variety of research purposes that work well, like storing inputs or outputs of research computing, being able to back up data, share it with other folks, and it meets data management requirements and compliance requirements. So it can be written in as the place to keep data while you're working on a grant before publishing it in a data repository. Another piece that complements ResearchDrive as well as other campus storage services is Globus. It's a web-based tool that makes it easy to transfer and share data between disparate systems. So at UW, we've got Globus enabled on several hundred different storage services or resources around campus. ResearchDrive is one of them, but there's also Globus connectivity at computing centers, like the Center for High Throughput Computing or the Social Science Computing Cooperative, as well as a number of research cores where data are generated, like the Cryo-EM Research Center, the Biotech Center, and a number of ones all over campus. So Globus allows you to be able to log in to a web interface, then be able to connect and transfer data between different resources. So some example use cases could be getting data from off of an instrument and moving it back to your research drive, or taking data from your research drive or another storage system and moving it over for computation at CHTC or at School of Medicine's Platform X environment. There's a number of things that are interconnected across campus. So in addition to moving data and shepherding it between different resources, it can be used. Both of these services can be used for sharing data with collaborators. Research Drive works really great for sharing data with other campus collaborators. It's available with anybody with a NetID. You can easily add them and share. And Globus works really well for external people, because there you can share it with people's existing email addresses at their own institution. You don't have to get them a NetID or a VPN or go through those pieces for remote access for external people. And then lastly, I want to highlight that these two services can also work together with some of the data repositories that are available for campus. So the integration we're working on right now is with the Minds at UW data repository that Trisha mentioned earlier with the libraries. So that would allow you to have a publication and have a persistent data object identifier that's published with that. It would point back to the Minds at UW repository where you've got all your metadata and details on there, and then you can link off to Globus where the actual data will be publicly available and then available for download for larger things. With that, we can accept larger data sets, currently on the order of a terabyte or things like that. So there's some stuff that can fit in there that aren't available at all the other sites. All right, and with that, we'll move on.
Hi, I'm Xinrui, and I'm an undergrad working with Professor Banks and Professor Barry Wanmin, and today I will talk about how we identify the hidden states of consciousness during sleep using multivariate vector autoregressive or NVAR models. First, I'd like to thank our lab members, our collaborators, and our funding sources. So during sleep, humans transition between different states of consciousness, such as wake, dreaming, or unconscious. Previous studies have shown that the traditional sleep stages represent mixtures of the actual consciousness state. So for example here, during the slow wave sleep, humans sometimes have no conscious experience, and sometimes they are having conscious experience through dreaming. And also previous studies have shown that during different sleep stages, the brain shows different patterns of the electrophysiological activity. And even within the same sleep stage, the brain can still show different activity based on the actual consciousness state of the subject, such as whether the subject is dreaming or unconscious. However, identifying those consciousness states is still an open question in neuroscience. So the question for us is, how can we identify the actual consciousness state of the subject during sleep using human intracranial electrophysiological recording labeled by the traditional sleep stages? And so to answer this question, we hypothesize that there are 4 consciousness states during sleep: CC+, which is connected to the environment conscious and alert; CC-, which is connected to the environment conscious but drowsy; DC, which means dreaming; and UC, which is unconscious. So to approach this problem, we first segment the whole night's recording into short 4-second segments. Then we cluster those 4-second segments using what we term the sequential multi-state NVAR algorithm. In NVAR models, the value measured in each channel at the current time point is approximated by the linear regression of values from all channels in the k previous time points. The sequential multi-state NVAR algorithm clusters those 4-second segments based on the distance between the data segment and the NVAR models representing the consciousness states. We initialize the NVAR models, which is kind of the cluster center here, by assigning sleep stage wake to CC+, sleep stage N1 to CC-, REM to DC, and N3 to UC. We then use a k-mean-based iterative reassignment strategy in which the distance is measured by the likelihood between the data segment and each NVAR model. This approach enabled us to identify the subject's consciousness state throughout the night at very high temporal resolution. The fraction of consciousness state for every sleep stage resulting from our approach is consistent with the existing studies about sleep. The effective connectivity change across consciousness states generally supports our prior hypothesis about the neural signatures of each. For example, that the temporal frontal connectivity here is higher in CC+ than in DC. Initial results indicates our algorithm has promise for identifying the hidden states of any system that can be reasonably approximated by NVAR models. And I'm happy to talk about how we can apply this method to the specific data that you might have that has kind of an internal connectivity that changes based on the metastable states of the system. Thanks.
Hi, I'm Matt Christie. I'm a data engineer at the Gibbs Land Use and Environment Lab. And today I'm going to talk about some of the work that we do to support the research that Holly Gibbs was talking about earlier, which is particularly studying cattle properties to avoid deforestation in the Amazon. So the work I do is often referred to as matching. And why do we do it? So we have some data that describes cattle movements in the Brazilian Amazon, has a source and destination, location or property, but those aren't spatially described in the data. There are other data sets that provide property boundaries or geographically explicit locations, but there's no kind of universal scheme for how to link those two. So the effort there is, in order to provide that spatial coverage for the cattle movements, we need to sort of develop our own methods for making those links so we can have the coverage on the ground. So describing matching a little bit, I mean, generally the high-level goal for matching is you want to be able to compute the real-world objects that digital records refer to. In our case, it's often like Excel, but that can be, like images or sound or anything, basically. But we often deal with human-generated data, which is very, very messy. There's lots of different ways to express the same thing, right? Oftentimes in our work, we encounter abbreviations, typos, other things like that. But there's other kinds of variability that you might have to address. So this process can be a lot. It's often very holistic, but one place to start is to find pairs of records that you think will plausibly refer to the same thing or plausibly match. So today I'm going to show you some work that we do for finding matching candidates for farm names. So one way we do that is we are able to take the names and map them into a high-dimensional space where we take those points and then are able to stretch the coordinates of those points based on how meaningful a given axis is. So here the axes are little bits of the string called trigrams, and we're able to weight those according to how meaningful we think that trigram is to reference a thing. So here you can see on the bottom, these are synonyms for farms. They don't really contribute much to distinguishing two farm names from each other, but there's kind of what we might refer to as the meat of the string, which have very, very highly weighted trigrams here. And so matching methods will kind of be looking for similarity among those substrings. And here you can see what that looks like. So this is a network derived from that process. So projected down to 2 dimensions for visualization here, but these edges are candidate pairs that were generated from closeness in this space. And so here you can see it's accounting for some of those synonyms, fazenda versus sitio. There's some variation in phonetic spelling, or even some more kind of egregious things down here that we're able to capture with these methods. All right, thank you. I am very passionate about this work, so if you're interested at all in entity matching or data integration in general, please find me after the talk. All right.
An exoplanet is a planet orbiting a star that is not our sun. There are a number of different methods for detecting exoplanets, and one popular approach is the radial velocity method, which looks for a wobble in a star. So as a planet, like this blue ball, orbits its host star, this red ball, from the point of view of Earth, we try to observe this forward and backward motion from the star. As the star is moving towards us, the light it emits gets blue shifted, and as the star moves away from us, the light it emits gets red shifted. And so if we estimate the radial velocity of a star across time and plot those radial velocities, if a planet's present, we'd expect to see something that's like a sinusoid, like the sinusoid curve we have here. So we have radial velocity on the vertical axis and phase or time on the horizontal axis. All else equal, a more massive exoplanet would have a higher amplitude, so a stronger signal. But astronomers would like to find low-mass exoplanets, such as Earth analogs, where Earth is considered a low-mass exoplanet. And this is challenging for a number of reasons, one being that the signal is very small. But another challenge is that the stellar noise can be periodic and look like a planetary signal. So on this graphic here, we again have radial velocity estimated against phase or time, and it looks like a sinusoid signal. You'd think that this was a planet, but in fact, this was just due to a star spot. So previously, our spectrographs were not stable enough or high enough resolution to detect the very tiny shifts that would be present due to these low-mass exoplanets. But more recently, the spectrographs have become more stable and more precise, higher resolution, and so these tiny shifts are now available to us. And so a spectrograph is going to collect the light, distribute it across wavelengths, and if onto a CCD, like what we have here, a charge-coupled device. And if we look at one row of this, we can see that we have what a function that is characterized by all of these narrow, some deep, some shallow dips. So you can think of the vertical axis as the amount of light, and the horizontal axis is wavelength. And so these dips or absorption lines are what help us to estimate the radial velocity. One nice feature is that if a planet is present, all these absorption lines will get redshifted and blue shifted together. But when it's stellar activity, such as star spots and other sort of features due to magnetic activity, that noise can produce shape changes in these lines. And the shape changes, though, will vary by line. And so you can think of the goal here as trying to separate these right and left shifts from amplitude or shape-related shifts in order to get the right and left planetary signal distinguished from the shape-related or amplitude changes due to activity or stellar noise that can either hide our planetary signal or produce a signal that looks like a planetary signal. So there are a variety of techniques that have been used and developed to address this challenge, but no clear solution has been found. New ideas are welcome. Thank you.